focus on the metabolomics approach. So basically, we will focus on 2 mini review for the method to watch. Okay, so first section, I would like to show you some tools for metabolomics analysis. And in the second section, we are going to talk about the annotating unknown metabolites. And that is published in this early year in Method to Watch. Okay. So in the first section, we will talk about the tools for metabolomics. So before we get into this section, I have a question for you. Okay, so do you have any concept about the metabolomics? Or can show me some definition of metabolomics or metabolome? Does anyone have any idea about this one? No? Okay, that's fine. Hopefully, after this section, you will more clearly understand about this metabolomics and metabolome. Okay, so metabolomics basically is a scientific study and that focus on the chemical processes of metabolites. So in other words, metabolomics is a systematic investigation that focus on the metabolite chemical fingerprint. And this chemical fingerprint is left after the specific cellular response. So which means that metabolomics is a fingerprint after all of our cellular response or biological response. Okay. So in contrast, this metabolome that is defined as all metabolites collection. So in the other words, these metabolites can represent as the ultimate product of the gene expression in that cell biology entity. So hopefully that definition is clearly for you to understand all of the content that we are going to talk about is for the metabolites that can basically recognize as a chemical fingerprint after specific cellular response. Okay, so let's look into this figure. Okay, so this figure basically is a central dogma of molecular biology. So hopefully you are familiar with this central dogma of this molecular biology because it describes that in our cell bodies, all of our heredical information are present in the cell as a form, as a DNA. And this DNA can be transcribed into RNA. And this RNA can be translated into protein. And all of this information can represent or modulate the ultimate function of metabolome. Okay, so which means that if one day or if at some conditions this DNA or this RNA cannot reflect the real situation or the real physiological activity with your cells, we can use the metabolome provide the snapshot of that condition within the cell. Okay, so that's basically reflect why we need to understand the metabolome, because metabolome in another approach, they can provide some information that's about what happened within the cell. Okay, so basically, I cannot understand your face activity. Is it clearly that what I say, or all of you are come from another field and do not understand what I'm talking about?
Oh, we understand.
I'm sorry?
Yeah, we understand.
Oh, you understand? Okay, so if you understand, please smile or not. Or I just feel confusing that what I'm talking about, okay? Okay, so basically now we know that metabolome is important because it represents the ultimate product of the gene expression in the cell entity. So basically, metabolomics can provide a better understanding. They can help us to understand the state of the cellular or the biological processes in different stages, such as when you are diseased, you have some disease. So at different stages, you will have different metabolomics analysis data. And sometimes you can get a better understanding under some disease condition. Okay, so since we know that metabolomics is a better understanding for us to figure out the cell response. So metabolome is represent the most closely representation of the phenotype. So based on this definition, so that's why we try to understand or try to determine multiple approach. They can help us understand what changed of metabolites in our cells. So not only for this paper, actually it's for multiple papers already demonstrate that the metabolomics analysis can help us to identify some useful metabolites. For example, because the previous study showed up that there are some 2-hydroxylglutarate upregulated in some cancer patients, such as the head and neck squamous carcinoma cancer patients. So based on this finding, there are multiple development focus on 2-hydroxylglutarate inhibitor development. So after we found this metabolite increase in the disease, we developed the inhibitor, and then this developer will be help us to treat in cancer patients. So theoretically, understand a metabolite can help us to cure cancer patients in clinicals. So that's why it's so important, and multiple approaches try to improve the finding of metabolites. Okay. So we already know that how important of metabolomics and determine the metabolites. So for the next, we want to understand that how to determine metabolites. Okay. So does anyone have any idea that how to determine metabolites? So the most common way, the most common method for metabolite determination is using this mass spectrometer approach. So this mass spectrometer approach basically divides into 3 parts, as shown here. The first part is the ion source, and the second part is for the mass analyzer, and the third part is detector. So we will get started into the detail that how metabolite can be determined through this workflow. Okay. So the first step, the metabolites will be ionized. Okay, so if the sample was ionized in the ion resource, and this metabolite will get charged, so it will become a charged ion. So when this charged ion shown here, you will find that we can use different force to separate this charged ion. For example, within the mass analyzer, we can use electric field or the magnetic field to separate this charged ion. Okay, so after in this mass analyzer for separation, we will move into the last step of detection. So what does the detector work with this system? This detector can detect each of the charged ion, and then they will transform the information of this charged ion, including the mass to charge ratio or the retention time. So based on this information from the charged ion, you will show up, there are different peaks show up in different time course and shown as the mass to charge ratio. Okay, so basically this is how we determine metabolites using this mass spectrometer analysis. And there is one point I would like to mention about here is about the the liquid chromatography and the gas chromatography. Since we put our ion in the, we put our sample in the ion resource, and we basically it's for different phase. We can use the gas phase or the liquid phase to help the sample on the column for separation. So that's some of the different kind of approach that we are trying to use in these systems. Okay, so basically we understand how does mass spectrometer works. So for the next, I want to show you some example that we will find after mass spectrum analysis. So this is one example of cocaine. So you will find out they are multiple fragment peak, and it shows us the different mass to charge ratio or the retention time. And basically, we will call this fragmentation peak as the spectrum. In Chinese, it's 光谱. Okay, so we will take this peak as a spectrum. And this is another example for this aspirin. Okay, so you will find out there are different spectrum, different fragmentation of aspirin compared to the cocaine. Okay, so which means that different compounds have different fragmentation pattern. So this could be help us to use the different pattern of spectrum data as a unique fingerprint for each different compound. Okay, so more than the application of this spectrum data as a fingerprint, we know that each molecule have their unique mass. So since this data is based on the mass or the mass to charge ratio, so basically we can through this approach to identify the molecular identification and confirmation. So that's basically how we determine metabolites and why we can use this approach for our topics today, that tools for metabolomics. Because different compounds have different unique mass, have different unique fragment, have different unique spectrum. So we can use this to identify different compounds and confirm the different compound. Okay? So this paper comes from the Nature method in 2021. So this is basically the workflow of metabolite determination. Okay, so you can find out that there are multiple steps, including the sample preparation or sample separation. Okay, so we will look into this detail. Okay. So basically, when we try to do this metabolites, we need to collect our samples. And since we mentioned about that metabolite is complicated, it determines different physiological response. Therefore, they have different pattern at different stage or different time posts. So which means that it's complicated. If you are unable to harvest your sample at the same conditions, it may probably reflect different physiological phenotype. So in other words, if today you want to get a good resolution, if you want to get a reproducible data that can reflect specific conditions, you probably need to focus on the sample preparation more carefully. For example, you probably need to follow the author's suggestions, including to avoid the environmental perturbations. In other words, if today you want to get a more reproducible data, you need to harvest your sample as much as you can at the same conditions. Okay. In another point that the author pointed out is about the enzyme activity. So if today you want to harvest your sample after the enzyme reaction, you need to make sure that you already stopped the enzyme activity, or your sample will have some effect from the enzyme, not the real situation within your cells. And the author also mentioned that in the sample preparation, we probably also need to include some standard spike in your experiment. So after this sample separation, we will move to the sample separation step. So the sample separation step is as we talked earlier. So you will find out that this sample will put on a column for separation. Okay, so you will find out it will separate in the condition here. So the same with we talked earlier. These samples will be ionized, and then ionized sample, which means the charged ion, can be separated within the mass. And then using the force, the physiological force to separate each of the ion samples, you will use the detector to get the information, including the rotation time or the mass to charge ratio. They can reflect the peak at different time posts. Okay. And the author also gave us some suggestions when you are working on this metabolomic extraction. For example, because the metabolite is complicated, so we probably need to at least take 4 biological replicate or add some technical replicate. Okay. And the author also mentioned that if we are working on a large scale study, we probably need to consider to add more quality control or have the batch correction in our samples. And after this sample preparation, separation, it's moved to the last step. And in the last step, they also mentioned that we probably need to focus or take care of some point about the ion separation or some the peak misunderstanding. Okay, so that might be happened in the last step of this the mass spectrometer analysis last step. So that's basically the Nature method suggests us to take point in our experiment. Okay, so now we basically understand that we can use the mass spectrum to reflect our metabolites. Okay, so basically when we try to identify our interest sample, for example, if I want to test our blood sample, okay, so at this time we usually take the mass spectrum data of our sample and compare with this standard spectrum library. Because in the condition with the standard spectrum library, we can match which peak is for which sample. In other words, if we would like to annotate metabolites, we need to rely on the matching spectrum data with our reference spectrum data. Okay, so this is the major point that we need to focus in each of our mass spectrum data analysis. And although we talk about that this approach can be used for determining the unknown metabolites, but they still have some point that we need to focus. And the first I would like to talk to you is about the library data. And actually, when we get started for this metabolite analysis approach, we usually need to compare our sample with the reference library. And the well-known library in this platform, probably in this metabolic field, is for this HMDB and methylene, these two libraries. So these two libraries basically can provide us some useful information for metabolomic analysis. But even it's a well-known library, reference library, more than 90% of metabolites in this database have no mass spectrum data. So which means that the challenge in this field is we do not have the standard spectrum data to compare. So we do not have enough metabolite spectrum data that can be used for us to compare our interested samples. Okay, so the limitation in this field is there are a higher percentage of the peak remains unidentified in the mass spectrum experiment. And there is another limitation in this field is about this. Another limitation is the accurate statistic validation for large-scale spectrum assignment. So when we are doing this multiple omics data analysis, we need to make sure that we get the correct positive sample and remove the negative sample. So when we are working on proteomics data, we usually create one artificial library. So this artificial library can help us to predict this false discovery rate. So we will know that what is for real, what is not for real. But when we are working on metabolomics field, because metabolite is too complicated, so we are unable to create a set that is available to predict this false discovery rate. So which means that we do not have a good tool to calculate what is real after annotation. So to solve this issue that we do not get a good approach for this false discovery rate estimation, the paper from the Nature Communication in 2017, they published one algorithm. So this algorithm combined two approach. One is for this empirical Bayes approach, and another one is target decoy approach. So this empirical Bayes approach is a common mass calculation, so which has been used in many computational analysis fields. And this target decoy approach, that is a common approach that has been used to control the false identification, because this target decoy approach actually uses two libraries. One is for real library and one is unreal library. So since we already know what is real and what is unreal, so we can, using the unreal to predict the false discovery rate. So the author used these two approaches to provide a good calculation that can help us to predict the FDR. So basically, this approach is based on almost 17 public metabolomics data sets to get this algorithm. Okay. So theoretically, we can using different improved approach to get the FDR, which might be helpful for us to annotate the compound that we are interested in. Okay. So, except for the improvement in this FDR estimations, they also have some progression in the compound identifications. So I would like to show you for this platform, CSI finger ID. So this CSI finger ID published in 2015, and this platform basically use this fragmentation tree and combine the machine learning and to predict which compound might be the real compound that you are looking for. Okay, so let's look into what it works. So basically, they have 3 steps in this platform. The first one is the learning phase. Okay, so in this learning phase, what they did is they used the mass spectrum data from the reference library, and then they generate this fragmentation tree. And then they use this fragmentation tree to predict this fingerprint. Okay, so this is in the first stage for learning. So after this system learn how to predict this fragmentation tree or the fingerprint, they will move to the prediction phase. So in this prediction phase, you can upload your mass spectrum data of your sample, which means the unknown sample you are really interested in. And then you can compute this fragmentation tree and then predict the fingerprint. So because we already have learned how to predict this fingerprint, and we also get the prediction of our samples. So in the last step of scoring phase, we can mapping our predicted fingerprint with this library. And that is one of the approach we can get our compound from the library. Okay. So this approach has been upgraded in 2019. Okay. So in this paper published in Nature Method, you will find out that the author incorporating the CSI finger ID platform in the Series 4. So this upgraded Series 4 platform combines not only the fragmentation, the spectrum and the fragmentation, they also include the structural data. Okay, so theoretically, according to the description of the author in the paper, they believe that this Series 4 can provide a fast computational approach for the structure identification. So for the next, I would like to show you how to use this platform. Okay, so this is basically what it looks like in the website if we get started using this Series 4. So in this first page, you can upload your spectrum data here. And then you can assign the conditions such as the ion condition that you are using in your sample for this data. And then you get the compute. So after compute, you will find out they are one item match with this system. So which means that this spectrum data is probably matched with this item. So there's only one item. But if we upload another spectrum data here, and the same we assign the condition that we are using, and you will find out that there are multiple potential targets, which means that all of these probably similar with your data, your mass spectrum data, your samples. But you can find out that there are multiple score points. So which means that the higher score probably represent the higher similarity. So this is one of the criteria, okay? Probably here. This score is represent that you can get more confident in annotating this as your unknown samples. So except for this prediction for this molecular based on our spectrum data, we can also look into the structure of this compound. So you can see here, there are different similarity of different compounds. And of course, we usually believe that the 100% of this prediction probably more similar or more likely of our metabolites. Okay. So basically, this Series 4 is a special tool. So they can basically answer two questions. Okay. The first question is, what is the molecular formula? And what is the potential structure? They can best explain our metabolite with the database. Okay. So now we probably know that they are new tools that can help us to predict the unknown metabolites. And in this paper, I would like to show you another concept about the isotope labeling. And as we know, the plant is have many different compounds that can be used in biotechnology or can use as the marker in disease or such like this part or reflect the response of cellular physiological phenotype. Okay, so the author gets started to identify the metabolites using this isotope labeling approach. So you can find out that they are using the carbon label the nitrogen label, or they are using the sulfide label in the living organism. So after label this isotope in living organisms, we can extract the metabolites from this living organism and get start the mass spectrum analysis. So basically in this paper, they also try to analyze the metabolites and the lipid, and of course they get a good result. And this is another platform that published in 2019. So basically this platform, they use 10-step, the chemoinformatics approach, and also they combine the isotope labeling. So what they did is they implement their data set in this platform and labeling and get star analysis. So they do believe that their platform can facilitate this carbon number identification, determination, and this metabolite classifications. So including, except for what we talk about, that there are multiple new approach to get the compound identifications. Isotope is another approach that can help us to identify the new compound or the new metabolites. And this is another approach I would like to introduce you is about this metabolomics. So this metabolomics is basically an integrated platform. Okay, so this platform, they use different modules, and this different module can help us to identify different patterns about metabolites. So the first module is about the mass spectrum data for the pathway identification. So if we use this module, we are able to identify the pathway activity that is highly correlated with your metabolites. Okay. And we can also use the biomarker module to identify the biomarker that is highly correlated with your samples. And there is another new tool for Network Explorer module. This module basically can integrate your metabolomics data and transcriptome or proteomics data set. So the same, I would like to briefly show you how to use this tool. Okay, so this is basically the website, the web page from this platform. And this platform is already upgraded to the version 5. Okay, so from this version, you will find out that you can process your raw data of mass spectrum data. Okay, and you can also identify the pathway based on your metabolite change. And also you can identify the biomarker or the other so many different tools. Okay, so for example, if we demonstrate you about the joint pathway module. Okay, let's show you this today. So basically, this module is try to integrate your metabolite data with transcriptomics data or proteomics data. So you can see here at this column, you can upload your gene, which means the transcriptomics data, or you can upload your protein, which is proteomics data. And in the right panel, you can upload your metabolomics data. And then if you get star analysis, they will show you some joint pathway that are probably in both of transcriptomics as well as the metabolomics data set. So this is one tool that can help us to integrate different data sets that probably we are interested in. Okay. So except for the identification or integrate different platforms, this platform can also upload different data with different form, such as you can upload your data with concentration, or you can upload your data with the peak intensity. Okay. And after you upload this data, you need to assign that which group is your experiment group and which group is your control group. So after that, they will help you to understand what might be the most change metabolized in your sample compared to your control group. Okay. So after assign the group, after the grouping, we will get start the data filtration. So this step is try to remove some peak that is not possible to be used in this module. Okay, so we will, based on their calculation, they will remove some peak that's not meaningful. And then we will get started normalization, because this is try to adjust your systematic difference among samples. Okay. So after that, you will get your data. And this data can be presented in multiple forms, such as you can present your data as the fold change, or you can present your data as the volcano plot, or you can also present your data as heat map. So there are multiple different formats that you can get from this the metaboanalysis analysis. Okay, so basically you will find out that in this data set, in this volcano plot, you will find out what metabolite was increased and what metabolite was decreased in your sample, of course, compared to the control group that you assigned. Okay. And you can also identify some biomarker. So this is the biomarker and with the statistic difference or with the different fold change. Okay. And you can also identify some enrichment pathway in your samples. So this is basically that we can identify all of this data from this platform. Okay, so basically this platform is an integrated metabolite data set. You can use this approach to find some metabolomic change in your sample or in your conditions. Okay, so that is another platform that has been published in Nature Communications in 2019. And this platform uses a network-based annotations, and that is focused on the metabolic reaction. Okay, so let's look into how it works. So we call this platform as the metDNA, the metabolized annotating and dysregulated network analysis. So in this platform, the concept of this paper is try to define one reaction pair. So you can see here, in the normal condition, we probably familiar that in the chemical processes, we usually, I should say that in a reaction, we probably will get the substrate, and this substrate will be catalyzed and convert into the product. Okay, so at here you will find out that the substrate A plus V is catalyzed into the product C plus D. And what the author did is they defined the reaction pair as the substrate A with product C here. So this is a pair of reactions. So the major concept, they define the reaction pair and create the network. And why they can use A and C as a reaction pair is because the A and C have the similar spectrum data. So which means that they have the similar molecular structure data. So based on this idea, they create this substrate metabolite and product metabolite as a reaction. Okay, so based on this one, they can create a network, and then you can upload your sample and based on this network reaction and predict what might be generated in your samples. Okay, so this is one tool that we don't need to have a real spectrum data because this information is come from the prediction of this network. So we do not have a real spectrum data here. Okay, but even we do not have the real spectrum here, we can also predict the potential metabolites in our sample according to this algorithm. So the author think that the improvement of this metDNA approach can expand the metabolite annotation, and then they can facilitate the integration of this multi-omics data analysis. Okay, so now we basically understand that multiple metabolites is significant in different disease or in different biological response. Okay, and now they have multiple papers showing that this metabolite can modulate the diversity processes, including the stem cell differentiation, the oligodendroid maturation, or the immune response. Okay, so based on the significance of this metabolite in determining the diverse physiological response. So that's why we use a whole section to understand how to identify the metabolites. And this tool from the 2018, they use one tool for metabolomics activity screening, MEX screening. So this database basically integrate the metabolomics data with pathways. So when we are able to identify the metabolic pathways, we are able to reflect the real cell response. Okay, so this is one of the tools that can help us to test our metabolites in the functionality in altering the phenotype. And this approach is basically carried out from this platform. And this platform is very similar with what we talk about the metaboloanalyst. Okay. So basically, in our first section, we talk about the different approach. So probably through this introduction and the different tool introduction, we know that the conventional approach in metabolomics analysis is for mass spectrum data analysis. But when we are using this traditional conventional approach, there is a limitation because over 90% of the peak is artificial, and this peak is not significant, not meaningful. So that's why many groups try to create a different approach to analyze, to annotate different metabolites. Okay, so we do believe that there will more and more data, more and more platform that can help us to process the metabolomics data set. Okay, so this is the first part of our course. So does anyone have question about this part? Yes.
The narcotic, is that the right word? Do they use, for example, police confiscated some drugs? They want to identify what it is. Is that what they go through with this kind of analysis, mass spectrum?
I'm sorry, are you talking about the samples?
Yes, if there are some unknown chemicals
yes.
that they confiscated.
Okay.
How can they tell what it is?
Okay.
Fentanyl, cocaine or whatever?
Okay, so basically this determine is, I think that I can use that cartoon figure to show you again. Okay. Here, so you are asking if I have one sample and I was unable to match with different peak, so how to determine the metabolite? Is it your question? Okay, so which means that I need to have the standard curve here, and then we can get understand what is metabolized. For example, if I get the product 8 that only match here but not here, so it might be not identical with aspirin. But if I get the product B, which have the similar pattern with the aspirin, I can probably predict that this product might be come from the aspirin because the metabolite is similar with aspirin metabolites.
So that's what they use?
There are multiple approaches. Some approach using this, but these have the limitation because, which means that I need to have the standard. I already know what it looks like, and I can get what I that's not possible to analyze all of the compound in the world. So the limitation is we do not have enough standard reference to reflect your metabolites. So new approach try to use different algorithms to predict the potential metabolites. Yes. So I talk about this approach, and this approach. This is all to identify some metabolite that do not have identical spectrum data.
But that's prediction, or are they validated by real?
Some of the data validate this data, but not all of them. So that's why we say that all of these metabolite analysis should be validated if you are really ready for food or ready for therapy. But that's not possible to validate all of these compounds. So they, for example, the paper, such as this paper, they validate probably 1 or 2 compounds and found that the validation of their sample is identical with this prediction. So they mentioned that this prediction probably work. Yes. So, yes.
Okay, the slide.
Yes.
The spectrum, the mass spec slide show only have pretty much just 1 compound.
Yes.
But if you collect the metabolite from us, the cancer cell, then they have million metabolites, then the peak will be very complicated.
Yes.
How do these organisms?
So that's why they need to consider so many algorithms, and they need to have a good false discovery rate prediction, because this false discovery rate can help us to remove some unreal, impossible metabolites. But I will not say that this approach is good enough for every single compound, because for our understanding, there is no approach that is far from perfect. So, but we still need to encourage this platform, because when we get started using this approach, we can find out some weakness point, and this weakness point can help us to improve the more sophisticated tool as established. So I would say we don't know exactly get a precise metabolites in our samples from this data set, because we do not have enough data. But they try to use different algorithms or different calculations to create one more potential prediction model for everyone to be used.
How about the quantity?
Quantity.
Yeah.
So you mean that how many samples I need to upload?
The one that they compare normal cells and cancer cells.
Yes.
Like compound A increased above in the cancer cell. How accurate can it tell?
Okay, I think that it probably depends. But if you would, if I think that I'm not sure of the platform, but in my experience, I have used this platform. So what I did is around 10 million cells for metabolite extraction, and this sample have their own control group. So I can get some information from this data set. But how quantify of these metabolites, I have no ideas. Yes.
How to determine whether to use gas chromatography or liquid chromatography?
I think that the gas and the liquid chromatography is in the phase that you firstly send your sample into the column. So it's just the approach for that one.
So I can use the same sample in 2 different approaches and I'll get 2 different sets of data from the same sample.
I think that you should get one sample in one column. That's not possible that after gas separation, you also put that in another separation. That probably doesn't make sense.
No,
right?
no, I mean like same sample we use in 2 different mass spectrometry, 2
Yes.
different units. One person is using liquid chromatography, another. So what kind of
Effect.
which one would be more realistic?
Oh, I see. Okay.
So there must be an advantage and disadvantage of using.
I see. Yeah. So you are asking which one is more accuracy for the mass analysis. And I have no idea actually. Sorry, probably need more experience can give us some answer, but does anyone have any idea about this technique? I have no idea actually, I'm sorry. Yes?
I have another question.
Yes?
In page 21.
Page 21?
A single compound. A single compound can have multiple peaks, but
Yes.
I don't understand why a single compound with one molecular weight can give multiple peaks. Because based on my understanding of proteomics,
Yes.
the fingerprint of one protein show multiple peaks because of the protein can be cleaved by tracing, and each fragment have different molecular weight. So I don't understand why single compound can give multiple.
I think that single compounds still have multiple components inside this. The single compound doesn't mean that they already have one unit inside. And they try to validate each of the components inside at different time course. So we call this for retention time. And then detect at different retention time what is their mass to charge ratio. So that is reflect this the data. That is the interpretation. Not like that one compound will only have one patent. Because one compound may have multiple components inside your compound. So that's my, what kind of component, what does it mean? For example, if how to say, if in a cell, okay, as the Dr. Lin mentioned about the cancer cell, if there is only one single cell there, and this single cell have amino acid inside, have lipid inside, have carbohydrate inside. So these 3 units in one cell will represent different peak at different time course.
So one component reflect
within one cells, there are multiple information can be detected because this multiple information detected is based on their different mass to charge ratio. So you will get multiple peak there.
This gives probably the even charge status, even though the mass is still the same, but charge is different.
So are you talking that we need to add one samples using different charge to isolate?
Automatically will have like one positive charge, two positive charge, they will have two there.
Okay.
Some case. So yeah, metabolite. What's the definition? Is that if I say degraded protein, does that count as metabolite?
You mean these words or these words or
Metabolite. I always confuse them. Is that only refer to small chemical compound or even nucleic acid, lipid, protein, or everything can be?
I think that all of this is inside because if you get one cell, that's not possible to distinguish multi-component inside there. Yeah, that's my understanding.
For example, the right-hand side show a chemical compound because I can see its structure, but it gives several peaks. So I don't know why this single chemical compound can give multiple peaks. If you are talking about a spectrum of metabolism, which is a collection of several metabolites, that I can understand why that kind of data can give multiple peaks, but I don't understand this case.
You don't understand this case, but this case is the same with previous case. Why I probably still not get into your point.
I think the question is the same compound, you have one peak.
Yes.
You say. In another way, the same compound has multiple.
No, one sample will have multiple peak. and multiple peaks represent the fingerprint of the samples. So it is not possible that one sample is only one peak.
Okay.
Yes. Okay. Thank you for interpretation. Okay, so do we need to have a break? Or we can, okay. Okay, that's 10 minutes break. Okay, so we will get our second section. So this method to watch is focusing on annotating unknown metabolites. And this is published in this early year. So in the first section, I think that we already know multiple approach that could be used to determine metabolites. Yeah, but there is far from standard reference that we try to identify our unknown samples. So the method to watch in this section try to talk about how to use the computational approach because this computational approach can push in the limit of unknown metabolite annotations. So in the second section, I would like to try to introduce some computational approach that has been applied in this metabolomics field. Okay. So before we get into our sections, I have a question for you. So I think that most of you are familiar with the generative artificial intelligence, right? So does anyone can show me what it is? What is AI? All of you should use the ChatGPT, right? No? Okay. Even no. Do you have any idea about the generative artificial intelligence or the machine learning? Does anyone can give me some response about this part? Do you know what is AI? Okay, you smile. That's better than nothing. Okay, so let's take a look into this definition of the generative AI. So generative AI is a technique. So they can use the artificial intelligence or the algorithm or the large language model to generate the content. And this content can contain text, video, or the picture, or any other non-existent content. Okay, so theoretically we can define that generative AI that use a large scale and use the different algorithms to analyze our data, and then we can use the data to mimic the style or the structure in our data set. Okay, so that's the best concept for the generative AI. And what is machine learning? Machine learning is a technique that has been used to help computer to learn tasks or actions. In other words, when we are using the machine learning, we usually put a large database, and then we force the computer to learn information from this big data set. Okay, so this is kind of definition of machine learning. So in more specific, we can recognize that the machine learning is a tool to get the generative AI. In other words, the generative AI comes from the result of machine learning. Okay. So when we are talking about the machine learning, there is another term that frequently show up with the generative AI. You probably hear about the deep learning or the neural network. Okay, so basically, this deep learning is a kind of machine learning. So it's a evolution approach of machine learning. So the difference of the deep learning is they use multiple interactions and then learn and then force computer to learn at different structure or different layer. So which means that they can learn multiple content and get a more optimized prediction model. Okay. And this neural network is also a method for the generative AI. Okay, so using this neural network learning, you will find out that it's kind of similar with our neuron in our brain. So they use a similar neuron approach to learn, to force computer to learn the information from the data set that we assigned. Okay, so that's the basically some terms that frequently show up in this generative AI field. But if we want to give you more familiar example, I would say the AlphaGo or the autopilot of Tesla, 就是下围棋的那个 AlphaGo 跟那个特斯拉的自动驾驶 that all use this tool of the deep learning and neural network learning. Okay, so since we know that this generative AI have a tremendous improvement in different fields, so that's why now we use this AI tool in the metabolomics study. Okay, so the watch to method in this section is try to show us how to use different computational approach to get a good annotating for the unknown samples. Okay, so this is the first example I would like to show you today. Okay, so this paper comes from the Nature Biotechnology in 2021. So basically, this approach uses the systematic classification of the unknown metabolites. So that include the different machine learning tool, and we call this as the canopus. Okay. So this is the workflow that I would like to introduce you about the canopus. And this figure is very similar with what we talked earlier. At first, we need to get the data set to learn, and then we can have a prediction according to what they already learned. Okay. So in this canopus approach, they have two machine learning models. The first machine learning model is try to force the data set to predict your molecular fingerprint according to your spectrum data. So basically, they will pull the spectrum data and compute the fragmentation tree and force the computer are able to predict this fingerprint. And another set in this canopus system is another machine learning data set here. So in this second learning model, what they did is they put the fingerprint as an input of the deep learning network. And then according to the input of fingerprint information to calculate the compound classification. Okay, so this is basically the canopus integrate these two machine learning tools for the compound prediction. So after force the system to learn these two models, we will get started for the prediction phase. So you will find out in this prediction phase, you will have your own mass spectrum data of your samples. And then you can use this system to compute your fragmentation tree and get the fingerprint. And then you will match this fingerprint with the learning data and get the potential compound classification. So this is basically the concept of this canopus system. Okay. So I would like to show you some data that the author provide to let you know that how powerful of this approach. Okay. So the author mentioned that this canopus can predict up to 90% of accuracy of the compound prediction. Okay. So what they did is they test some sample, and of course they get a higher prediction. So they mentioned about this part. But one of these tools have a tremendous improvement in identifying the metabolites that did not show up in the library. So in other words, what they did is they removed the compound of this flavonoid glycosidase. So they removed this compound, and then they retrained this model. This model already have no flavonoid glycosidase. Okay, but even they remove the mass spectrum data of these ones, they found that this canopus data can also predict the compound of flavonoid class. So which means that this machine learning already upgrade to extend their compound classification, not only based on the spectrum data. Okay, so this is one of the powerful function that the canopus data have. They can predict the compound class even there is no training mass spectrum data there. Okay. So except for this tool, this powerful functions, they also provide another functional analysis. It's about the metabolite determination of different kind of classes. Okay. So when we are working on this metabolomics analysis, we are really interested in identify the metabolomic change in our conditions. Okay, so which means that if we can provide a platform that can identify your metabolite changed in our sample, that would be very useful and powerful. And they also found that their approach can get this aimed. So what they did is they reanalyze the mass spectrum data from another group. And that data is comparing the germ-free and specific pathogen-free mice. So after reanalyze this data, they identified the significant change metabolites in dataset comparing the germ-free and specific pathogen-free mice item. So all of this is up to the 10-fold statistic difference in their prediction. So based on this data, they mentioned that this canopus tool is very useful in identifying the metabolite change among different conditions. Okay. And they also mentioned that this canopus tool can predict the molecular network. Okay. And molecular network is also a very popular method. They can help us to pushing the search boundary and get the potential metabolites under the spectrum data. Okay. So what they found is this color means that the spectrum data already in the reference hit. Okay, already in the reference library. Okay. And this green color means that that is not in the reference. It's based on the structure. Okay, so they get this one. And these canopus try to link the different compound with different similarity. So you can see here, if the light is for solid light, which means that it's have a higher similarity. But if the line is for dot or the dash, which means that it have the lower similarity. So they also mentioned that based on the canopus predictions, they can get a network. They can show the connection between two compounds. Okay, so based on these calculations, they think that they can also calculate all of these as a boundary of one kind of compound, such as the phenolic glycosidase. And this boundary shows the compound classification of isoflavonoid. And also they have other compounds that can be predicted in this canopus network. And they also found that even they was unable to find a compound with any connection with this work, they can also predict this compound in the class of isoflavonoid, because this compound color here is an aqueous compound, and this is the downstream of the daisy, and daisy shown here. So they also think that even we do not have direct connection of this new compound aqueous with other compound, but we can also predict the compound classification. So this is one of another tool that they also mention about in this Canvas tool. This Canvas tool can also help us to predict the chemical diversity. Okay, so you can see here, this 4 is for different euphorbia subgenera. And this table shows in these 4 subgenera of euphorbia, they have 6 different compounds. And what they found is, if you look into the compound diterpenoid, if you look into this compound diterpenoid, they have a higher activity in the species of this Ischia, and also higher activity in this subgenera of euphorbia. In contrast, if you look into this triterpenoid, this subgenera have a higher activity compared to this and this. Okay, so based on this data, they also find the other compound classification distribution in different euphorbia species here and here. And you can find out they have different components. So they also mention that this canopus can identify the diversity patterns among different species, subgenera of plants. And they found that this may provide a new version of the evolutionary tree identification. Okay, so this is kind of another approach that can help us to discuss the evolution of this plant. And this canopus tool can also used for the structure identification. So you can find out that this tool combines 2 platforms, including the classifier ontology and this series analysis. That is what we are talking about in our first section. And this classifier ontology is a kind of system prediction classification of chemical compound. Okay. So combine these 2 platforms, they also found that this canopus can help us to identify the new compound, because what they did is extract the mixture from one species, the Rivularia species, which is a kind of marine cyanobacteria. And when they extract this mixture from this species, they try to analyze the structure and the mass spectrum data. And they found a similar or primary component in this mixture. And that is very similar with the deoxypeptide. So what they did is they add more tool about the NMR tool and dissect this compound. And they found a new compound named as the cyclodeoxypeptide. Okay, so this is one example that the author mentioned that we can use this tool to identify the new component, new structure from our metabolite mixture. And they also have other approach that can help us to identify the unknown metabolites using the machine learning tool, such as the DAC, NPS, or the mass novelist that has been published last year. Okay, so you will find out that there are much more tools using the computational notation to determine the unknown metabolites. Okay. So this is another example that I would like to show you is about the net ID. And this net ID, they use another alternative algorithm to predict the compound. So let's see how it works. So basically, this net ID use another algorithm. We call this for integer linear programming tool. And this tool has been used in other fields for the optimization analysis. So it's a kind of math, but it has been used in different type of field for the optimization. So they use this in metabolomics tool. Okay, and basically this approach include the same 3 steps, including the candidate, annotation, the score, and the optimization, right? Network certification, optimization, yes, optimization, okay. So what the major concept in this net ID is they use different algorithms for the integer linear programming. And then using these algorithms, they define each of the peak from your mass spectrum data. And then they define each of the peak as a node. In addition, except for this node, they also defined the connection between 2 nodes. Okay, so we call the connection between these 2 nodes as an edge connection. And this edge connection is basically generated by adding or removing atom from the node here. For example, if we add the atom of sodium and H from the node A, and this would be the node A plus sodium and proton. Okay, so this is the concept about this approach. They create the original seed, then matching with your spectrum data here. And then they create the potential connection using this edge connection concept. And in these edge connections, they also include 2 types of prediction connections. One is for biochemical connections, and another is abiotic connection. And this biochemical connection is basically derived from the metabolites. So they identify the connection from the real metabolites. And this abiotic connection is derived from the mass spectrum phenomena. So it's a kind of real metabolite and a technical peak from your data set. So we can take some example to figure out the connections. So for example, if there is a difference of the 2 protons, and then we will define this connection as a biochemical connection, because this connection may be derived from the processes of oxidation or reduction relationship. So it was defined as the biochemical connection. In contrast, if the difference is from the sodium and proton difference, we will suggest this is by the abiotic connection. And sometimes this connection will be established in both of biochemical or abiotic connection. For example, if there is a water loss or water addition, and we know that if the water loss is caused by dehydration, and they might be derived from the enzyme activity. So you lost the water, or it might probably due to the mass spectrum signal. So in these conditions, the water loss can be defined as both of biochemical connection as well as the abiotic connection. Okay, and they also define another node as the putative metabolites. And this putative metabolites is basically a formula. So this formula was not found in the data set, but there is a connection to the metabolite. So there is additional node that has been used to define your network. So this is basically, they generate different C node from your mass spectrum data. And they also generate the yellow node from the connection data. And they also have some edge connections, which show the potential connection of your samples or your prediction. So this is all the concept about this network in this net ID approach. So based on this approach, they create almost 25 biological connections and 59 non-biological connections. So based on this, they develop a unique algorithm for us to predict your unknown samples. Okay, so after define such as the network, including the edge connection or the C node or the metabolite, putative metabolites, they also use some unique algorithm to optimize these conditions. So I would like to show you one example that how they show the optimization in this tool. Okay. So this is the prediction, the example that they also try to demonstrate how powerful for the optimization. Okay. So you can see here, basically they have two nodes, A and B, and they define as the AMP and adenosine. And since they also find 5 peaks in these connections, so they include all of these 5 connections. So based on this edge connection and the C node, so they predict 2 potential networks. One is left, one is right. So let's look into how they predict the compound based on this node. Okay. So in the node C in the right network, you can see here, the node C is annotated based on the node B with HCl. So this prediction should be for this one. And this is for the right network in here prediction. And in the right panel predictions, they predict the node C from the node A with the CO2 loss. So based on different connection, they have different compound prediction. Okay. And finally, they do the isotope labeling. So after the isotope labeling, they will find out that the node D with an additional carbon labeling here. And the node E have additional chloride isotope labeling here. But if we look into the right network prediction, it's only for carbon prediction here. There is no prediction here. But if we look into the real C, you will find out that because only the node C in the right panel have the chloride here. So this compound should be derived from here, not here. There is no chloride here. Okay. So based on this prediction, so they also find that the canopus prediction here, which means that here have a higher score. And the right panel here have a low score. So they predict this is for the real annotation in your unknown samples. So this is kind of the logical that this canopus tool try to annotate an unknown sample. Okay. So they also confirmed the network availability in yeast or in the mouse tissue. So they found that compared to the mass spectrum data only, or the node score only, or the node plus age score analysis, this net ID optimization can get a lower false discovery rate, which we talked about earlier, that this can really reflect the accuracy of your samples. In contrast, this net ID optimization approach can get a higher accuracy compared to the other algorithm using only one condition. Okay, So which means that, yes, this canopus is powerful to get a good annotation of your unknown samples. And this tool also have another improvement in the metabolite derivative identification. So let's look into this yellow node. So this yellow node means that this compound show up in the formula, but not in your library. It show up the connection, your biochemical connection, but not in the library. Okay. So based on this canopus prediction, they find 3 diamine derivative from this diamine. Okay, so you can see here, this is diamine, and this is diamine plus oxygen. This is diamine plus C2H4O, and this is diamine plus C2H2. So after predict this connection, what they did is they analyzed the mass spectrum data here. So you can see here, this is the diamine spectrum data here. So you will find out they have 2 peaks. One is the major thiamine, and another one is the unmodified thiazole. But if you look into the derivative of thiamine here, you will find out they have the similar peak compared to the thiamine, which means that it's derivative from this thiamine. And they also found that this derivative have additional peak, which is this structure with the acetylation pattern. Okay, so it's a kind of derivative, not identical, but have additional modification. And this another thiamine derivative is here. You can find out that they have a major peak here, but there is no unmodified thiazole. And in this derivative, you will find out additional modification in here and here. So this mass spectrum data shows that indeed this canopus tool predict relative putative metabolites, and that is real from the thiamine derivative. And they also use the isotope labeling to confirm this data. So what they did is they feed the yeast with the carbon label glucose in the conditioned media, and they found that the thiamine and the thiamine derivative have the labeling, the 13 carbon labeling. But if we use some unlabeled thiamine in this conditioned media, you will find out there is no labeling of the isotope. And since we can get some prediction of the thiamine derivative from this canopus tool, but if we look into the real situation, you will find out that this derivative is actually uncommon. In other words, the authors are curious about is this a real derivative or is it artificial? Does this have really meaningful for some physiological condition? So to understand where is this derivative come from, so they try to understand the potential reactions that come from the conversion of thiamine into the thiamine derivative. And what they found is this pyruvate dehydrogenase can catalyze the thiamine into the thiamine derivative. Okay, so which means that the derivatives is actually show up in real world, not just the computational prediction. And except for this pyruvate dehydrogenase, they also come from different potential enzymes, including this alpha ketoglutarate dehydrogenase. Okay, so using this alpha ketoglutarate dehydrogenase, they can convert the substrate of ketoglutarate into this substrate, into this derivative of thiamine. So which means that it's really happened, not just from the computational annotation. Okay, and the other enzyme can also come from this substrate to become a diamine derivatives. So they also think that net ID can help us to identify the derivatives which might be not show up in your previous metabolomics data set. So which means that they can get a more broad implication in this field in the future. Okay, so they also use this cartoon figure to summarize what this tool functioned. So basically, they found that this net ID can use this integer lineage, this integer linear reprogramming to create more network identifications that can help us to identify the unknown or that has never been annotated before. Okay. So I think that the major concept in the second section is they also point out that indeed we can use multiple metabolomics. We can use multiple computational approach to improve the identification of the unknown samples. And although this is far from the perfect, but we do believe that we will get a more quality or real annotation in the field. Okay, so this is basically our second part in this course. So do you have any idea or any question about this part? Or you already have all the questions in the last class? Actually, the second part is kind of similar with the first part, but it's just re-emphasize that in this field, we already get started to involve more computational approach to identify the new metabolites. Even I'm not exactly understand how to do the calculation, but the point is how we can use this approach in our research project. Okay. So, yes?
May I ask about the input data for the neural network system?
Which system?
The input data, so they are using like the mass spectrometry
Yes.
database?
Yes.
And so they are all using only...
All of the talk that we are talking about basically is using the mass spectrum data. But the difference is we have different algorithms to generate different library reference. They can help us to matching with our samples, our unknown samples. But all of this is based on the mass spectrum data. Yes. Oh, yes, that's perfect. Okay, so please email me if you have any question. Okay, thank you.